Observing of an embryo, such as the chick, helps us to understand the development of a mammal, like ourselves. A fertile hen's egg contains an embryo and enough food to enable it to grow, develop, and hatch into an independent animal. In this film, we open a series of incubating eggs to see some of the steps in this process.
After incubation of a fertile egg at body temperature of the bird, which is approximately 102 degrees Fahrenheit, hen's eggs are opened carefully with forceps so as not to injure the embryo. In the earliest stages, the embryos are taken from the shell and photographed under a microscope in culture dishes. Here, the size of the embryo in relation to the shell is shown after 18 hours of incubation. Through the microscope, we can see the embryo already beginning. A dark streak vertically through the middle of the oval lighter area. Cells that have multiplied and piled up cause the darkness of the streak. About 6 hours later, by 24 hours, the anterior part of the brain is forming and can be seen at the upper end. Behind this, the notochord forms a thin, dark streak down the middle. 4 pairs of somites have formed and show as dark blocks on each side of the notochord. These will form muscle and vertebrae. Time-lapse photography, condensing the growth of 3 hours, in 25 seconds shows the early formation of the heart. Large hollow vesicles come together from the sides to form the heart between them in the midline. The embryo at 33 hours has a clear area around it, but outside that is a ring of forming vascular tissue that will grow out to cover and enclose the yolk, forming a yolk sac that will develop blood vessels to bring food from the yolk to the embryo. A close-up of the head of the embryo shows the beginnings of the eyes, optic vesicles that grow and bulge out from the sides of the brain. In the middle of the embryo, we see the posterior part of the heart. The 2 large veins coming together to form the posterior of the heart are the vessels that come into the heart from the forming yolk sac. Behind the heart, somites can be seen. The embryo now has 9 pairs of somites, and usually the heart begins to beat at this time. The heart has grown more rapidly than its surroundings, so that it bulges out to the right. The beat is at first irregular or spasmodic. After 2 days of incubation, the embryo is this size in relation to the shell. Removed from the shell, the embryo can be seen to have grown in length and flexed so that the head is bent over in a C-shaped curve. The anterior end of the embryo has also rolled over onto its left side so that we see only the right side of the head. The heartbeat has now become rapid and regular. A close-up of the heart through the microscope shows the squeezing action of its muscles as it propels the blood forward into the arteries of the body. Slow motion shows that although valves have not yet developed, the squeezing action of the heart muscles keeps the blood moving forward. Out on the yolk, the vitelline blood vessels have formed to bring food back to the embryo. Blood cells can be seen coursing through them, some going away from and some coming back to the embryo. The arteries carry blood away from the body to the yolk, and the veins carry blood back to the embryo. At 4 days, the embryo is this size. From this time on, embryos will be photographed in place on the yolk in the open shell. On the side of the head is the right eye, darkly pigmented, with a light area in its center, the pupil. The wing bud is the white area just below the beating heart. In the tail region, we see the allantois, a large saccular outgrowth from the hind gut. It has many blood vessels in it, and it grows extremely rapidly, filling all the space between the embryo and the yolk sac on the inside and the shell membrane on the outside. Combining with the chorion, it becomes the respiratory organ of the embryonic period. Through the shell, its blood vessels exchange oxygen and carbon dioxide with the outside air. Eventually, this combined extraembryonic membrane, the chorioallantois, will have more of its surface occupied by capillaries than there is left between capillaries. Our own lungs are like this also. The heartbeat is shown in slow motion. The embryo now begins to increase rapidly in size, and by 7 days looks like this. It is now in rapid motion because another extraembryonic membrane, the amnion, which surrounds and protects the embryo, contracts spontaneously and rocks the embryo back and forth, thus preventing adhesions of the membranes. The eye shows clearly, and below it, the light areas are the wing and leg of the right side. The large blood vessels coming up from the embryo are the vessels of the allantois that carry the respiratory exchange gases from the shell. At 10 days, the embryo has formed beak and claws. There are now reflex movements of head, wing, and leg. The eyelids begin to grow closed at this age, but they open again before hatching. The egg tooth, a specialized horny structure, grows on the upper tip of the beak. It is used only in helping the chick to break out of the shell at the time of hatching. It is made of material similar to that of our fingernails and drops off some days after hatching. At 13 days, the embryo is this size. Down feathers show clearly. The weight of the embryo has caused it to sink into the yolk, but it is also using the yolk up more rapidly now. Leg and foot movements can be seen. At 19 or 20 days, just before hatching, the beak of the chick is thrust into the airspace at the large end of the shell. The chorioallantois, the respiratory membrane with its many blood vessels, is peeled back so that the chick can be seen more clearly. The chick remains in this state for some time, breathing a little air that diffuses in through the shell at the blunt end. And sometimes the chick can be heard to peep inside the shell at this time. Then a reflex action of the chick, lifting the head backward, causes the egg tooth on top of the beak to cut the shell, making a small hole. Poultrymen call this pipping the shell. The chick remains in this state for 6 or 8 hours, or in some cases even longer. During this time it breathes, develops its lung function, and begins to cheep frequently. The chorioallantois now begins to dry up. It is no longer needed for respiration. When the chorioallantois is dry enough, the chick is free to rotate within the shell. With a backward thrust of the head and an outward thrust of the legs, it begins to turn a backward somersault within the shell, rotating itself, with the egg tooth cutting the shell bit by bit. In half an hour or so, it cuts almost around the shell in a circle. When it is cut far enough, the thrusting of the legs will cause its body to push off the cap of the shell it is cutting. The chick emerges very weak and wet. Inside the shell, we see there is still a little blood remaining in the vessels of the chorioallantois lining the shell. Excretory wastes that have collected in the allantois during development are still clinging to it. But as the chick pulls free of the last bit of the allantois, they will be dropped. A few hours later, the chick is dried and fluffy. The down feathers keep in its body heat and protect it. The chick will soon begin feeding for itself.